So the final one on these little videos about structure of some of the chemicals in biochemistry is going to look at amino acids, and I'm just going to address a little bit of a confusing topic first of all. So the basic structure of an amino acid is this. Nitrogen, carbon, carbon, that's the backbone if you like. And then if we fill the rest in, we've got the two hydrogens on here, that will be the amine group. I'm going to have a hydrogen here. Now this is our carboxyl group. That's the carboxyl group at the end. And I've got an R group sitting off there. Now, depending on what you look at, you will also see, or you can also see it drawn like this. So amine group on the end, fine. There's our backbone, CC. And I'm going to put the double bond down there this time. You might see it with the R group instead flipped around. And you know which version of these is right. The answer is it doesn't really matter. It's probably easier to use a model. So this here is a model of an amino acid. So here's our NH2 or amine group at this end. There's the carbon, the hydrogen. I've used this to just represent an R group, and there is our carboxyl group on the end. Now, because molecular structures, these bonds here, they're free to move. Okay, I can twist these things round, so I can change the shape of it a little by moving these bonds around. Now, why would it be one way and not the other? It would depend really on what this was. So we know that the R groups are all different, and they have different properties. Some are more positively charged, negatively charged. Some have got more hydrogen bonding going on. Some might be able to form out disulfide bridges and so on, but polar and non-polar. And basically what a molecule will do, it'll find its most stable configuration. In other words, it will adjust itself around until the atoms are happy with themselves, I guess, is one way you can think of it. So it doesn't matter if you draw it like that, or you know, if you want if you want to have the R group on the bottom, fine, we'll move it that way. Oh, there we go. It doesn't matter, you know, the thing can move around. Now, one point here I think is worth remembering, that R group, if you draw the amino acid, I'm just going to do it quickly like this. Okay, so it's the same thing, I've just not drawn all the bonds. If I put that H there, that is the amino acid glycine. So an R group, which is simply hydrogen, is glycine, and it's an easy one to remember. It's useful because you can give it as a specific example. Okay, if you ever are asked to draw structures out, you can now draw a genuine amino acid rather than just putting the R group, which is can be one of 20 things. Just put an H on there, it's suddenly glycine. Okay, so that's a useful one. The only other bit that's, in fact I've got one here, there's glycine, now two hydrogens. The only other bit that causes problems, I guess, is when you come to the bonding, peptide bonds. And there's a few little odd things that happen here. I'm just going to mention a couple of them. Now, amino acids, usually when we draw them, we draw them like this. But when they're in the body, they're actually in water because they'll be dissolved in stuff. In fact, what happens to this carboxyl group, the reason it's acidic is it donates its hydrogen off. And in fact, what happens, a bit oddly, is that this bond between the two oxygens, it kind of flips between like that. It's called a resonant bond, and it just sort of flips between these two. So it actually forms something quite stable at this end. So sometimes you'll see the carboxyl group at the end written like that, and it just means it's lost it's lost that hydrogen off the end. Why will it do that? Well, it depends on the acidity of the solution it's in and all kinds of stuff. Anyway, in fact, it often gets picked up on this end, so you'll sometimes see this written like this. And that'll have a plus charge, it picks up a proton, but it's actually all the same stuff. Usually we stick to this in biology, so at A level, just in case you see these structures popping up anywhere and you think, what on earth is that talking about? That's what it's talking about. So let's just reform these for a second. So the peptide bond is the only other thing really to know about. Again, if you've watched the other videos on structure, you'll know that the thing to look for is always OHs. Okay, this is going to be a another condensation reaction, so we want an OH and an H from here in order to make water. So I'm going to pull that off there, and we'll pull that off there. Pop, pop. There's my water from my condensation reaction, and I'm going to join my carbon and nitrogen. Now you can see here the way the way it just formed. Those two oxygens have sort of come close together, and in reality it doesn't like it. So when these amino acids are being put together, it also depends on what these R groups are again. The structure of it is actually determined by some of these bonds that can move. In fact, this one here doesn't rotate very much at all. It's pretty stable, it stays where it is, but the others can. And when you're talking about the tertiary structure of a protein, the idea of these amino acids forming, as they form, they will bend and form shapes specific to what these R groups are. That's what's happening at the kind of molecular level. So just if we're going to draw that out, we'd have H2NC, let's put our R groups on the top because we are. I'm going to put, I'll draw up my full bit. This is going to bond onto my next amino acid, like so. So I'm looking here, there's my OH, let's take those two apart, and the bond, I'm not going to redraw all of it, but once I've got that backbone, NCC, NCC, what have I got left? Well, I must have a double bonded oxygen sticking off the top, and the rest is just going to be, there's a nitrogen there, and there is my peptide bond between the two. Of course, I'd still have the R groups, which side it goes on. I think by convention it's easier to have my double bonded oxygen on the opposite side of the R group, but again, it doesn't matter. It depends on what this R group is, really. So that's entirely up to you how to do it. Don't worry about it, is what I'm trying to drive at, I suppose. And one last point, excuse me, on proteins and polypeptides, since it's some times comes up. If you jam a couple of amino acids together, you could call it a dipeptide, because there's two of them, a bit like disaccharides. If you have a bunch of them together, you can call it a polypeptide. What's the difference between a polypeptide and a protein? Nothing really. Usually these are referred to when you're talking about things like enzymes, complex hormones, which are big proteins. You'd usually use the word protein. Sometimes when people are talking about simpler ones, maybe only a few amino acids long, they maybe just call them a polypeptide or a peptide. To be honest, they're the same thing. It's not something to particularly worry about. That sounds a bit more scientific, doesn't it? But yeah, don't worry about which one you use. It is effectively the same thing as far as we're concerned.